Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. For decades, scientists, pathologists, they've seen that our cells command center the nucleus. It often gets these really weird shapes in diseases like cancer or, just as we get older. But here's the question. Are these wrinkles and funny shapes just symptoms? Or are they actually causing genes to switch on or off? Well, today we're diving into a breakthrough that really for the first time gives us a kind of microscopic GPS to see exactly where and how this nuclear shape messes with our genes.
That's exactly right. We're digging into a really fascinating paper from Science. It's called Expansion in Situ Genome Sequencing, Links Nuclear Abnormalities to Aberrant Chromatin Regulation. And our mission today basically is to unpack this new tech called ExIGS expansion in situ genome sequencing. It's pretty groundbreaking because it finally lets us eavesdrop on that hidden conversation between the nucleus's physical shape and what the genes inside are actually doing, especially when things go wrong, like in aging or disease.
Okay, yeah, let's set the scene a bit more, because for years, this has been a bit of a puzzle, right? Pathologists, they could look under a microscope and see these visual clues. You mentioned wrinkles, but also things like blebs or lobulations.
Yeah, signs that something's off.
Exactly, like cracks in a foundation. They knew it signaled problems, maybe cancer, maybe just aging. But they couldn't connect those shapes directly to which specific genes were going haywire inside those exact same cells. That link was missing.
Right, a huge gap.
And the tools we had, traditional genomics like single-cell RNA-seq or ATAC-seq, they got really good at listing which genes were active, which DNA was accessible.
Fantastic list, yeah.
But they lost the where. All that crucial spatial information inside the nucleus, it just vanished. You couldn't tell if a gene turned off because it was suddenly squished up against the nuclear edge, for example.
The context was gone.
And even the newer spatial methods, while better, often didn't have the zoom power, the resolution to see inside the nucleus at that really fine, base pair level detail. So for ages, it felt like we had these two puzzle sets, one showing nuclear shape, the other showing gene activity. And they clearly should fit together, but they just didn't.
Until now, yeah. That's where ExIGS comes in. It's designed specifically to bridge that gap.
Okay, so let's unpack ExIGS then, this microscopic GPS. How does it actually work? What are the key innovations that finally let us link nuclear shape and gene function with this kind of precision?
Well, the really clever thing about ExIGS is that it's not just one single invention. It's more like a fusion, a combination of three big technical steps, each designed to get around those problems you just mentioned. Think of it like a three-phase journey right into the nucleus.
Okay, phase one.
Phase one tackles the size problem. The nucleus is tiny, right? And lots of important stuff inside is sub-diffraction, just too small for regular microscopes to see clearly. So what ExIGS does is, first, they embed the cells or tissue in this special hydrogel. Then they chemically trigger the gel to physically expand, uniformly, in all directions.
Wait, they actually stretch the cell bigger.
Yeah, literally.
Yep.
Imagine taking a tiny blurry photo and carefully stretching it out, maybe 4 or 5 times bigger, so suddenly you can see all the details. That's kind of what they do to the cell structure. It makes those tiny, previously invisible features big enough to see with standard microscopes.
Huh. That's clever, but doesn't that mess up the internal arrangement?
That's the crucial part. While they expand it, they use chemistry to anchor the DNA and proteins to the gel matrix. So as the gel swells, everything stays in its relative place. The internal map, the 3D structure, is preserved, just magnified, no jumbling.
Okay, that makes sense. So physical magnification using a clever gel trick. That gets around the imaging limits. What's phase two?
Phase two is about knowing where each piece of DNA is within that expanded nucleus. This is the in situ library construction with spatially registered barcodes. So instead of pulling the DNA out, they chop it up right there inside the expanded nucleus. And then this is key, they add unique molecular barcodes called UMIs to each little DNA fragment.
Like tiny address labels.
Exactly, like tiny address labels. These barcodes capture the precise 3D coordinates of where that fragment was sitting in the expanded nucleus. So you get the location tag right along with the DNA piece. They can then do some initial sequencing steps right there in situ inside the sample to read both the 3D location and that unique UMI sequence. It's like scanning the address and a bit of the mail content on the spot.
Wow. Okay, so expanded view and now address labels on the DNA. What's phase 3? How do you read the actual genome sequence and put it all together?
All right, phase 3 is the computational back projection and multimodal integration. This is where the heavy data lifting happens. After they've got the location info in situ, they then take the sample and do standard high-throughput sequencing ex situ outside the cell to read the full genomic sequence of all those fragments, along with their UMIs.
Ah, so they get the full message later.
Precisely. And then powerful computer algorithms match the in situ 3D locations with the ex situ genomic sequences using those shared UMI barcodes. This creates essentially a nuclear atlas. It computationally places every single sequence DNA read back into its exact original 3D position within the nucleus, perfectly aligned with images of proteins, like the nuclear lamina, which they also capture. They even measure the exact expansion factor for each experiment to make sure the distances are calculated correctly.
So it's like you're mapping the entire genome, practically base by base, onto a 3D scaffold of the nucleus, and you can see exactly where proteins like lamin are relative to every gene. That sounds incredibly powerful.
It really is.
But they must have checked this really carefully, right? How do we know this expansion mapping process is accurate? That it's not introducing weird artifacts?
Oh, absolutely. Validation was key.
Yeah.
They showed ExIGS massively increased the number of genomic reads they got per nucleus from around 300 up to about 6,000. A huge jump in detail.
Almost 20 times more data per cell.
Yeah. And spatially, it sharpened up the protein imaging resolution significantly, down to about 220 nanometers. This let them see DNA-protein interactions much closer than before, like less than 200 nanometers apart, which was previously just a blur.
Okay.
And crucially, the biological validation. They showed that where DNA was close to known repressive structures, like the lamin protein or compacted heterochromatin, that DNA was less accessible, just as you'd expect based on standard methods like ATAC-seq. So the 3D organization it revealed matched known biology. It holds up.
Okay, fantastic. So the tech is validated. It gives this incredible detailed map. Now, the big question, what did they learn when they pointed this new nuclear GPS at a real biological problem, a disease?
This is where it gets really interesting. They focused on a disease called Hutchinson-Gilford progeria syndrome, HGPS, or just progeria.
Ah, the disease that causes rapid aging in children.
Exactly. It's a devastating genetic disorder caused by a mutation in the gene for lamin AC. This leads to a faulty, toxic protein called progerin building up. Now, normally, lamin proteins form this meshwork, the nuclear lamina, right under the nuclear membrane. It provides structural support, keeps the nucleus shape stable, and also helps organize chromatin, often silencing genes near the edge.
Right.
In progeria, because of that faulty progerin, the lamina gets messed up and the nucleus becomes misshapen. Using ExIGS, combined with its high-res imaging, they could watch this happen in fibroblast connective tissue cells from progeria patients as the cells aged in the lab.
What do they see change?
They saw the nuclear shape getting progressively more distorted. The lamin at the edge got thicker, but also they saw these prominent invaginations, like deep folds or buckles of the lamin extending into the middle of the nucleus.
So the inner lining of the nucleus was literally folding inwards, creating complex internal structures.
Precisely. And the effect on the chromosomes inside was striking. In normal cells, chromosomes kind of meandered around, but in the progeria cells, they saw the chromosomes starting to follow these weird internal contours of the buckled lamin, almost like the distorted shape was physically pushing the DNA around.
Wow.
They quantified this too. They measured how much of the nucleus volume was taken up by lamin, especially this internal lamin, far from the edge, and that percentage shot up dramatically as the progeria cells aged. They also confirmed it was mostly the faulty progerin protein making up these abnormal structures.
Okay, so the structure is clearly abnormal and gets worse over time. But the key link, did this affect the genes?
Absolutely. This is the core finding. They found that a staggering 98.3% of genomic regions ended up being closer to lamin structures in the progeria cells compared to healthy controls.
98%. Almost everything got pushed closer to the lamin.
Almost everything. And crucially, this increased proximity directly correlated with changes in chromatin accessibility. Regions that got closer to lamin generally became more repressed, less accessible, likely shut down.
So the distorted shape wasn't just passive. It seems to be actively forcing potentially active genes into contact with the repressive lamin environment, messing up their regulation.
That's exactly what the data suggests. The abnormal nuclear mechanics seem to be directly driving aberrant gene silencing.
So, okay, that's a huge finding for progeria. But was it just like a total breakdown inside? Did the whole nucleus just become disorganized chaos, or was it more specific? What were they looking for there?
That was a really important question they asked. Did these lamin problems cause a sort of global decay of all chromatin organization? Or did they create more localized hotspots of disruption, leaving other areas relatively normal?
Right, like specific bad neighborhoods versus the whole city falling apart.
Exactly. To figure this out, they developed this really neat analysis. They looked at small 2-micrometer spherical neighborhoods around each DNA red they mapped with ExIGS. Within each tiny neighborhood, they checked the relationship between lamin proximity and chromatin accessibility, using ATAC-seq data as a reference for what should be active or inactive. If lamin was near inactive DNA, heterochromatin, that's normal. They called it an organized neighborhood.
Makes sense.
But if lamin was found close to DNA that should be active, chromatin, that's abnormal, suggesting repression where there shouldn't be. They called those disrupted neighborhoods.
Okay, so they mapped the nucleus for these organized versus disrupted zones. What did they find in the progeria cells?
Here's the surprising part. They didn't see a global collapse. Even in the older, more messed up progeria cells, over 80% of the neighborhoods were still organized.
Whoa.
But scattered amongst them were these distinct, concentrated hotspots of disrupted neighborhoods.
So it wasn't total chaos, more like specific, localized problem areas.
Exactly. It ruled out the idea of a uniform decay. And importantly, they found a direct link. The more of that weird internal lamin a cell had, the more disrupted hotspots it tended to have. Those inward folds seemed to be the breeding ground for this misregulation.
Okay, so localized disruption linked to the internal lamin structures. Did these hotspots target specific types of genes, or was it random which genes got caught?
It seemed fairly evenly distributed across the genome, actually. The hotspots themselves didn't show a strong preference for particular chromosomes or large regions. But when they looked specifically at the genes located within the accessible parts of these disrupted hotspots, the genes that should have been active but were now potentially being silenced by lamin proximity, they found something interesting.
What kind of genes?
Genes involved in things like the actin cytoskeleton and focal adhesion, these are absolutely critical for how fibroblasts work, their structure, how they move, how they stick to things.
So genes important for normal cell function were getting caught in these lamin traps?
It looks that way. The idea is, if these normally active essential fibroblast genes get accidentally dragged into a lamin disruption hotspot, they might get aberrantly shut down. And that silencing could directly contribute to the problems seen in progeria cells, like them becoming too stiff or losing their proper shape and movement, basically, features of cellular aging.
That's a really compelling mechanism, linking the physical shape change directly to silencing specific functionally important genes via these hotspots.
Okay, this is fascinating for progeria, but is this just a weird thing that happens in this one rare disease? Or could this mechanism, this link between lamin shape and gene silencing, be relevant more broadly, like in normal aging, for instance? That's exactly where they went next. And yes, the implications seem much broader. First, they confirmed that lamin, whether it's the normal stuff at the edge or the abnormal internal stuff, generally acts as a repressor. They looked for RNA polymerase II, specifically the active form, POL2, SIR5P, which is the machinery that actually transcribes genes. And they found a clear depletion of this active transcription machinery near all lamin structures, both in healthy and progeria cells.
So lamin basically creates a no transcription zone around itself.
Pretty much, yeah, a no-fly zone for active genes. Then they expanded their view beyond progeria. They used ExIGS on cells from mouse hearts, both the muscle cells, cardiomyocytes, and other cell types, and also on fibroblasts from a 92-year-old human donor.
And what did they find in those normal aging or different tissue contexts?
They saw a whole variety of nuclear shapes, and importantly, they saw prominent internal lamin folds and structures in many of these cells too, looking quite similar in principle to what they saw in progeria, just maybe less extreme or developing more slowly. So they quantified this again, calculating that repressive zone, the percentage of the nucleus volume that's very close, within 200 nanometers, to any lamin structure.
Okay, what were the numbers?
Well, in young, healthy control fibroblasts, it was around 35% of the nuclear volume. In the progeria fibroblasts, as we said, it jumped up to about 64%. Now get this. In the fibroblasts from the 92-year-old donor, it was around 61%.
Wow, almost as high as in progeria.
Almost as high. And in the mouse, cardiomyocytes, which are highly specialized long-lived cells, it was even higher, around 66%.
So this isn't just a progeria thing at all. It suggests that as we age normally, or in certain cell types, these lamin abnormalities accumulate, significantly expanding the repressive territory within the nucleus.
Precisely. It points to this being a more general mechanism of gene regulation, tied to aging and cellular specialization, driving transcriptional repression. So the model they propose is quite elegant. Normal cells have a neat peripheral lamin layer, keeping specific stuff quiet. But with aging, accelerated or normal, lamin can accumulate, the nucleus shape gets irregular, you get thickening, and these internal folds. These abnormal structures then increasingly encroach on normally active chromatin regions, creating those local hotspots and leading to inappropriate silencing of genes that should be on.
This really does change the picture, doesn't it? ExIGS seems like more than just a new technique. It feels like a new way of seeing and understanding the nucleus. What really makes it stand out as an innovation, and where does this take us next in research?
I think what's truly groundbreaking is how it reframes the nucleus. It takes it from being this kind of complex, somewhat fuzzy organelle to being a quantitatively measurable 3D space where mechanics directly link to gene regulation, that pathologist's observation of a wrinkled nucleuses.
Yeah.
ExIGS allows us to potentially break that down into specific sequence-defined problem zones. So that's a massive conceptual leap.
From a qualitative observation to quantitative mapping.
Exactly. And technologically, it's that unique fusion. The expansion microscopy for super resolution, the in situ barcoding for spatial context right down to the DNA sequence, and the sophisticated computation to put it all back together. It delivers something quite unique. Base resolved genomic reads with their precise subnuclear 3D address. Other methods often have to trade one for the other.
And that combo makes it a powerful tool for testing ideas, right? Like how nuclear shape causes gene changes.
Yes, it creates a causal testing ground. You can see how the mechanics, the lamina buckling, relates directly to which genes get silenced nearby.
The potential applications seem huge. What about clinically?
Oh, definitely. You could imagine using ExIGS signatures alongside traditional pathology grading to get much more precise cancer diagnoses or assessments of tissue aging. Better classification could lead to better treatment choices.
And finding new drug targets.
Absolutely. By seeing exactly which enhancers or other regulatory elements get inappropriately shoved next to lamin when things go wrong, you might identify completely new targets for therapies aimed at correcting that misregulation. And even for testing therapies, you could potentially use ExIGS to see if a drug aimed at, say, improving lamin function actually succeeds in normalizing the chromatin organization locally and restoring proper gene expression. That's mechanobiology in action.
Beyond specific diseases, what does this tell us about basic biology, about gene regulation in general?
It really underscores that nuclear architecture, the shape, the organization of things like lamin, isn't just passive scaffolding. It's an active player in gene control. Variations in nuclear shape might be a much more important layer of gene regulation than we previously appreciated, relevant in development, health, and disease. And there's this intriguing idea that maybe these internal lamin structures could form partly in response to things like DNA damage. If so, that could be a mechanism linking cellular stress directly to gene repression, perhaps contributing to some of the epigenetic changes or increased noise in gene expression we see in aging.
And the technology itself is adaptable.
Yes, that's key too. They've already shown it works in rat and mouse tissues beyond fibroblasts, so it's a generalizable platform that researchers can now potentially apply to a huge range of biological questions and systems. It really feels like a launching point.
So as we wrap up this deep dive, it leaves us with a really provocative thought to consider, doesn't it?
Definitely. Imagine if every little change in your cell's nuclear shape, every wrinkle, every fold, isn't just a side effect, but an active dynamic force that's constantly remodeling how your genome can be accessed, literally dictating which genes can be turned on or off at any given moment. What could that deeper understanding mean for how we think about tackling aging or treating diseases where nuclear shape is known to be altered, like many cancers?
A truly fascinating thought to mull over. We hope this deep dive into ExIGS has given you a fresh and maybe slightly more structured perspective on the incredibly dynamic world inside every one of your cells.